Hey everybody, we're in the home stretch. We got 3 more weeks left of the semester. I'll be talking to you during a live session about the exam schedule. So the third week is going to be exams. We'll probably have a day to review as well that week. So I'll go over that a little bit during the live session. I'll also post the exam schedule on Canvas as well. So without further ado, we're going to jump right into this. We got about, like I said, 3 more sets of notes to do in the next few weeks, and we need to get going. So we're going to jump right into this. So we're switching gears now. We're entering Module 5, which is the last module for the semester. And this module is going to focus more or less on genetics. So we're going to be talking about DNA, RNA, and proteins. Today's focus will be focusing on DNA, and then we'll go over RNA and proteins a little bit later in the week, as well as the next week and we'll be finishing up Module 5. So we got 3 lessons to do. All right, so let's go ahead and we'll jump right into this. Module 5, Lesson 1. There's the learning goals. You can look through that and kind of get an idea of what we're going to be covering. I'll let you read through that on your own. It's also on Canvas. And today's focus will be DNA. So we're going to be talking about DNA, discussing DNA. We might touch a little bit on RNA and proteins as well as we go through this, because you got to kind of talk about them all together. But we'll try to keep our focus mostly on DNA today. And then the next couple sets of notes will be focused on RNA and protein synthesis and how that works. All right, so our first little section on DNA is going to be focusing on structure and function. We'll kind of split it up, and we'll go over the structure and then the function and talk about how it works, how it gets replicated, etc. And we'll just run right through this, okay? All right, so what is the function of DNA? So the function of DNA, basically DNA, the way I like to explain it to my students is it's the instructions on how to make proteins. That's pretty much it. Proteins determine all your characteristics. It could be metabolic characteristics. It could be physical characteristics. All these different proteins kind of control what physical and metabolic and different types of characteristics you have. And DNA is the instructions on how to make each of those proteins. So in this section, we're going to learn about the different functions of DNA. The main function of DNA is it's a molecule that contains the genetic information found in all cells, and basically what determines those characteristics that we see in different organisms are the proteins that they are able to make using those sections of DNA. It's found in all living organisms. It supplies the information necessary for cells to reproduce. It's responsible for determining how a person looks. That's those characteristics I keep talking about. And it has codes for the formation of proteins, which kind of control those characteristics. So we'll be going through all of that today, and then we'll talk a little bit more in detail about RNA and proteins and how that works as well later in the week. So it contains codes for the traits that make up organisms and those that are passed from parent to offspring. So we pass these traits on, or this code on how to make certain proteins on to our offspring. It contains codes for diseases that are genetically passed down from parent to child. And it's basically an important area for researchers who determine the role genes play in complex diseases such as cancer, diabetes, heart disease. And we're getting really cutting edge right now. There's a new technology out there called CRISPR technology, which is fascinating technology for me. And I might talk about it here and there as we kind of go through some of the genetic information. All right, so what is the structure of DNA? Now you've heard it called a double helix. I'm sure you've talked about it. It kind of looks like a twisted ladder or a spiral staircase, or however you want to kind of explain it. But the structure of DNA is very complex. Even though it was first discovered in 1878, we still didn't really know the genetic secrets that it carried or what it kind of looked like until the 50s with Watson and Crick. If you look at the picture to the right, it looks kind of like a twisted ladder. It's in the shape of what we call a double helix. This ladder has 2 main parts, and that's the sides. So if you take a look, you kind of got these sides. This one's winding around this way. The other side's kind of winding around the other side, and they kind of match up here in the middle, and we're going to talk about that in a moment. And it's pretty complex. It's one of the macromolecules, right? These side chains, right? These side chains are made out of alternating sugars and phosphate. Deoxyribose, which is basically where DNA gets its name, deoxyribose nucleic acid, That's the 3 letters. So it's a deoxyribose sugar, and then it basically has a phosphate, and then another deoxyribose, and then a phosphate, and then another deoxyribose. And these middle parts are the nucleic acids that we're talking about. That's the Na part of it. Okay. So the sides are basically create up the bone of DNA. They're made up of sugars, as I mentioned, and phosphates. And then the second part is the rungs of the letter, and that's created by the nitrogen bases. And they pair up in a very specific way. We'll talk about that later. But you can see here something called thymine pairs up with adenine, and then cytosine pairs up with guanine. And we'll talk more about those a little bit later as well. So how are DNA related to chromosomes? If you look at the image to the right, it shows various forms of DNA. DNA is usually this double helix, as we just mentioned, right? So you see this double helix. Well, if you take a look at that, if it were unwound normally in a cell, that's how it is, it's unwound, you wouldn't even see this molecule. It's very, very thin, right? It's a large, a really long molecule, but it's very thin, and you would be able to see it even with the best microscopes that we have out there as far as light microscopes. But then during cell replication, these things wind up, they kind of coil up, and then they start wrapping around these little proteins called histones, and they get super coiled, and they form these structures called chromosomes. And during times of replication, we can actually see the chromosomes. So when it gets super coiled into chromosomes, we can actually see this molecule, but normally when it's unwound, we don't see it at all. In the image, you start on the lower half, right, where it's labeled double-stranded, and you can see that, and that's kind of what I was doing with you. And then to the left, it starts to unwind, and it kind of gets into this, or it starts to wind up and gets into these histones, and then it forms chromatin, and then eventually it forms these chromosomes. Okay, so in order for chromosomes in DNA to be wound, they wind around those special proteins I just mentioned called histones. If you think of DNA as thread, then histones would be the spool that you're wrapping this thread around. Okay, as the DNA wraps around these histones, it also coils up into these tight spring-like forms, and this can be seen in the image right above the part labeled histones. And these tightly wound springs form chromosomes, which are in the X-shaped structure, which are called chromosomes. Right. So you have these chromatid forming these chromosomes, and then the chromosomes, you only see them when the cell's getting ready to replicate. Even in prokaryotic cells, it's kind of similar. You have it forming this basically circular piece of DNA called a plasmid. So essentially DNA makes up chromosomes in all living things. On the chromosomes we find in genes. Genes are specific locations for the codes of proteins that make up these characteristics. But then even in prokaryotic cells, bacteria cells, for example, the chromosomes aren't really called chromosomes. They're called plasmids. They're these circular, basically kind of, if you take a look, it's just this big long loop, and this one kind of loops around a little bit, but it's basically this circular ring of DNA that kind of bacteria uses, and it's a very simplified version of this chromosome structure. Our chromosomes are more linear, theirs are circular. So when we talk about DNA, we're referring to the genetic material all the time. So DNA was not always considered the genetic material. Matter of fact, for a long time, people thought proteins were the genetic material. They didn't realize that DNA is the instructions on how to make proteins. Matter of fact, they thought DNA was probably just this organizational stuff, and it didn't really have much use at all other than organizing the proteins in some way, shape, or form. And really, it wasn't just Griffith and Hershey, which are talked about here, and there's a great video you should watch on Canvas that kind of goes through their experiment. But it was a whole bunch of, it was a whole line of scientists over the course of decades that kind of was able to realize that DNA is actually the genetic material, and it wasn't the proteins that was the genetic material. So prior to Griffith's and Hershey's experiments, many ideas on what the genetic material was in living things dated all the way back to Aristotle. With the data gathered and analyzed from Griffith and Hershey's experiment, we know that DNA is responsible for containing the genetic information. So I encourage you to really watch that video. And again, if you did some research, you'd find out that it was actually a whole bunch of scientists even before them that kind of built up to their experiment. So why is replication important? Unfortunately, most of us have been injured at some point in our lives, right? We get a paper cut or scrape our knee or, even break a bone or do some kind of damage to your body. In order for your body to heal the wound, that new skin cells have to be created. So there has to be replication of our skin cells. To accomplish this task, the cells must first copy their DNA because each cell must have their own set of DNA, and then they replicate and they become new skin cells. Even human life begins as a single cell that was formed when the mother's egg fused with the father's sperm. The single cell then begins to divide. It goes through replication again. We have to replicate our DNA, and then the cell divides. Um, and as it goes through this series of mitosis, uh, we have to make copies of the DNA for each cell. So that single cell then divides, and then it still contains some of the DNA as the original, and the cell continues, and every cell has the same copy of DNA. Finally, you eventually get this multicellular, made up of millions of cells, trillions of cells, in the form of a human, or if it's another animal, some other animal or plant, or really any living thing um, that we know of basically has DNA as its genetic material. Another reason DNA replication is so important is inheritance. Inheritance is what you get from your parents on the way of genetic code, right? So each cell in the body contains 23 pairs of those chromosomes that we were looking at. One chromosome from each pair is inherited from your mother, and then one is inherited from your father. So if you think about it, you have 23 pairs of chromosomes. Half of them come from your mom, half of them come from your dad. If you remember when you learned about the structure and function of DNA, right, as we're going through this and we're talking about the structure and function, genes contain the coding of DNA which corresponds to a specific location on the chromosome. In order to pass those genes on to future generations, the DNA would have to be copied. If this did not happen, the passing on of genes would not occur. DNA provides the information for all living things. Its replication is basically essential for life to continue. So we have to have this replication kind of occurring. So to summarize some of the reasons DNA replication is so important: growth of organisms, right? Repairing damaged cells when we get an injury or damage to our tissue, passing on of the genes from one generation to the next, and basically it ensures all new cells have the instructions it needs to survive, to make the proteins it needs to do its job. All right, so let's look at the steps of replication really quick. DNA helicase, this is the multi-step. You're going to hear all these ACEs, okay? Let me preface this a little bit. So you're going to hear about DNA polymerase, ligase, primase, all these different things that end in ASE. If it ends in ASE, it's basically an enzyme. Think of these things as little enzymes that are doing jobs. And remember, enzymes are basically a type of protein. So these are little proteins that kind of do all this work inside the cell when we want to replicate DNA. So the first thing you have, and I'll kind of point these out, is you have this little enzyme, and this one's kind of represented by a triangle. This isn't really what they look like. This is an artist's rendition to kind of make it a little easier to understand. Um, they're not perfectly like this. They're proteins, right? So you have this one protein called DNA helicase that comes in, and it basically unwinds the double-sided um, molecule, right? It basically takes and unwinds the double helix. So the junction between the unwound part and the open part is called a replication fork. So it opens up this little replication fork where these other proteins are going to get in here and do their job. Then you have this enzyme called DNA primase. This thing comes in and synthesizes a short series of RNA nucleotides that are complementary to the DNA strand. So this primase comes in here and kind of gets it started. It primes it, if you will. It starts the basic little area where we're going to start putting together these nucleotides. And then you have DNA polymerase 3, which comes in and adds the complementary nucleotides and binds the sugars and the phosphate. And then you have DNA polymerase 3 travels only in one direction, which is kind of important. It travels from something called 5' to 3' end. The 5' end and the 3' end is based on the carbons on the sugar. And if you take a look at a sugar of deoxyribose sugar, you have these carbons, and they're basically numbered 1', 2', 3', 4', 5'. So when we're talking about 5 prime, it's the carbons on the left-hand side of that molecule, if I were drawing it on the board, and it moves from 5 prime to 3 prime. So DNA has a directionality. That's the important part you need to know. So, and then the next step, you have one side of the lead, it's called a leading strand. Okay, so this is the leading strand down here, and this is basically the lagging strand. The reason it's called the lagging strand is this one has to be built in sections. So basically you have to have this RNA primer kind of built in, and then it builds a little, because remember it has to go from 5 prime to 3 prime, right? So it builds a little section, and then it backs up, and then it builds another little section, and then it backs up, and then it builds another little section. So this one's always lagging behind. This one, it can build it and move in that direction in one swoop, right? It just moves right along behind helicase and kind of builds it in one continuous strand. So one side is the leading strand. Once the RNA primer has been laid down by the primase, the DNA polymerase thread can build on the leading strand and continue to build in that same direction and just kind of move right along. The other side is called the lagging strand, and that it's called the lagging strand because the helicase or the helix uncoiling occurs in the opposite direction. So um, it basically has to, the DNA polymerase has to basically work in section. It has to build a little section and then back up, and then build another little section and back up. And then you have this other organism come in called ligase, and it basically kind of, if you take a look, there's a problem, right? Polymerase enzyme reaches the end of the replication fork. It's not able to add any more bases since the helicase is moving in the opposite direction. So the solution is you have this polymerase bind further down the chain, and basically creates these things called Okazaki fragments. It was named after the scientist that discovered this, was a scientist named Okazaki. And basically it makes these Okazaki fragments, and these are basically kind of broke apart. So we have to have this other guy come in, DNA ligase, and he kind of welds these parts together. So if you look, here's an Okazaki fragment right here, right? And it's not connected. So we have to have ligase come in, and it basically, after the fact, kind of welds this together or puts that last strand or part of the strand in to make sure it's one continuous strand. Kind of crazy that all this is happening inside the nucleus of our cells right around the time that the cells are going to divide. So during replication, there's many points along the DNA that are synthesized at the same time, right? It's not happening just in one spot. It's happening in multiple replication forks. It would take a very long time to go in sequence from one end to the other. So it's much more efficient to open up several points, have several replication forks at one time. This type of replication is considered semi-conservative because each new strand is created by using the original half. It uses the original half as a template. All right, so now let's start to kind of briefly preview what we're going to move into in the next couple weeks. So far in this lesson, we've learned about the structure and function of DNA and how it replicates, but we also need to talk about how it has this code, okay? So we're talking about this genetic code. You hear this all the time. They talk about the genetic code, right? So it has this code, and in this section, we're going to explore why DNA is able to do so much. But first, let's review some facts that we need to kind of consider when we're talking about DNA. DNA is made up of nucleotides called adenine, thymine, cytosine, and guanine. I mentioned those way at the beginning, right? A always binds with T, T binds with A, and then C binds with G, or G binds with C, and I don't know why it's missing there, but I'll add it in. So you have these things always kind of binding together. So how is DNA used to build proteins? Well, it's basically built into this genetic code. You have these 3-letter codons, and we're going to find out that each of these codons code for a specific amino acid. And then when we read the 3-letter codons, we can put the amino acids together in the right sequence, and that sequence determines which protein is being built. So phosphate and deoxyribose sugar molecules create a backbone, right, for the DNA molecule, while the nitrogen bases are bonded together with hydrogen bonds at the rungs of these ladders. And that's the A's pairing with the T's and the C's pairing with the G's. Each chromosome is created by a really, really long DNA molecule that's tightly wound up. All the cells have the same set of DNA, and the DNA sequence is passed on from parent to offspring for all organisms. So if you take a look, and we look at this, you have these, as I mentioned, 3-letter codons, and we're going to go into more detail on this in a later set of notes. But if you look here, for example, you have GCA. Well, that's the codon, right? The genetic code for a particular amino acid called alanine, okay? AGA codes for arginine, and so on, right? So you get these three-letter codes. So as the three-letter codes are read, you have these other proteins during protein synthesis, which again we'll get to later, that strings together the amino acids in the right sequence, okay? So basically, the DNA structure, the sequence of these bases, determines the protein structure. So DNA is able to do so much because the different combinations or sequences of the nucleotides, the A's, the T's, the C's, and the G's, provide the code for building proteins. Basically, the genome, or the complete set of DNA for an organism, holds the instructions for amino acids, which are the building blocks of proteins. Remember, DNA is made from smaller molecules called nucleotides. Proteins can vary in size, but are basically built by smaller units called amino acids. These proteins are important because the proteins are the fundamental components of any organism. They determine how all the molecules are organized, how they act inside the organism, physical characteristics, metabolic characteristics. So therefore, the proteins play this crucial role in the way the organism looks, grows, functions, and reproduces. So if proteins do all of this, then why does DNA get all the credit? Well, again, as I mentioned at the beginning of the notes, because DNA is the instructions in how to build the proteins. That's because without the sequence of DNA, proteins would not be built, right? You wouldn't know, or the cell wouldn't know how to build each of the proteins. So DNA provides the template for the protein, and the proteins do the grunt work in expressing the code of DNA. In the picture that you can see in Canvas, it's basically, let me back up. It's this picture that we're talking about. In that picture that you can see in Canvas, the DNA sequence at the top is the genetic code that determines how the protein sequence is being built. So if I back up again, that's what I was pointing out. So this sequence right here is the code for each one of these. So you have a three-letter code for this amino acid, 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 three-letter code for this amino acid, and we continue on for as many amino acids as we need it for that particular protein. So we're going to take a much deeper look into exactly how this happens when we go into lesson 2 and lesson 3. We'll probably start lesson 2 a little bit later this week because this is a short week as far as assignments go. I didn't put any heavy assignments in here. I think there's basically a one of the science notebook assignments again, and then a little uh, um, assignment that you have to basically do a message board assignment. So that's it. So let's go over those right now. Make sure you do all the self-checks for Module 5, Lesson 1. Do all the checks for understanding quizzes for Module 5, Lesson 1. Remember, you can do those multiple times, so everybody should be getting A's on those. And as I mentioned, kind of a short one, a little 15-point DNA science notebook that's in here. That's Module 5, 1.5 assignment. And then make sure you do the Module 5, 1.8 discussion board assignment on human genome after you watch the video. And that's basically it, guys. Kind of a short week as far as assignments go. But we're going to jump right into the second lesson and go into a little bit deeper look at this later this week, probably on Friday for you guys. So once again, have a good day, and I will see you a little bit later, and we'll talk a little bit more about the exam schedule coming up in 3 weeks.